So we continue our journey in haptics with Domenico Prattichizzo. He's professor at Siena University, and he's also senior researcher at IIT, Italian Institute of Technology in Genoa. Domenico.
Thank you. So it's for me a pleasure to stay here and stay with all the organizer and you in this wonderful anniversary of our conference, ICRA. Okay, I will start talking about wearable haptics that I've been investigating over the years with excellent colleagues like Ellison and Catherine, who just presented their work before my speech. And then I will talk about a very new idea that we had in my lab that we are really enthusiastic about. So the first thing that I want to highlight is the wearable technology that has been already covered by, so the importance of wearable haptics is relevant because we went from something very similar to a robot on the left to something that is still a robot, like this mechanism that is a parallel mechanism that moves a platform under your fingertip and try to simulate a force at your fingertip, okay? So that was one of the first wearable haptic technology allowing to apply a force on the body and not a vibration, with several applications like teleoperation, rapid prototyping, VR, and AR. From that idea, we patented several concepts, including one that is collocation of vibration, skin indentation, and temperature. And this goes in a startup that I co-founded in 2018, that is WeArt, and this is our last technological engineered product that has 6 points of actuation collocated. So this was one of the first contributions in wearable haptics. More recently, I have been working on the concept of augmentation, where wearable haptics play a big role. This is within a project on human augmentation, where Tamim and Asfur and Nikos Tsagarakis are partner in Europe. Basically, the idea is to try to extend the human sensory motor system with the sensory motor artificial system of a robot. Supernumerary limbs, basically, okay? So I want to connect my body to an extra limb to do more advanced operation, but especially to help people with disabilities. Look, for example, in this case. In this case, there is a paretic subject who has a wearable device that gets the motion from the body and controls the extra limb, in this case, a human hand, and a robotic hand, sorry. And this robotic hand provides a feedback on the contralateral hand to close the loop between the sensory motor system of the human, biological, to the sensory motor system of the robot that is artificial. This is the example. By moving the hand, is closing the robot, and is now perceiving on the left hand, on the white band, with our wearable technologies, all the forces applied by the robot. Why we want to do this? We want to do this because the human should have the agency of the robot. I give always this example. For example, if I'm on a chair and I'm a paretic patient and I want to serve a drink to my girlfriend, for example, I want to do it. I don't want to have a robot doing it for me. I want to be engaged, even if the performance are lower than having a robot itself. So having the agency at the sensory motor loop with an extra limb is very important. Well, the last idea I want to talk about, still related with wearable haptics, is this one. So this is something very strange, because sometimes the idea comes where you meet people from very different fields. I met Serena Zacchigna 2 years ago, during the COVID actually. She invited me for an event in Trieste, and I was talking with her, and she said that she was studying why in the heart as organ there is no tumor. At least the tumor is very rare with respect to the other organs. And while talking, she said, Domenico, the reason is that because the heart is the only organ that beats forever. Okay, and this makes in some way the cancer going away proliferate less. You know that at that time, I from this concept of the heart, so a synapses in my brain started, and I get this idea. So the idea was this following. So I said, look, but I'm developing technology to stimulate the skin. Why I don't use this technology to stimulate, for example, a melanoma, and having this tissue behaving like an earth in terms of mechanical stimulation of the tissue. So that was the idea. So having a non-cardiac tissue with a cancer, for example, a skin cancer, a melanoma, and applying our technology to reproduce the heartbeat or heartbeat-like. This idea, of course, I'm here. It's very, quite simple, but quite interesting in the sense that, of course, if I am here, we are having very interesting results, and I'm going to show you the preliminary results that we got in this kind of research. I will start with the animal cancer models. There is a lot of animal models. In particular, in this mouse, we injected cancer cells in the left and right leg. Then we treated the right leg with the soft wearable haptic stimulator, simulating this periodic stimulation, and then we went to see, after a while, the situation of the cancer in both legs, control leg and treated leg. Some detail about the technology. The technology is pneumatic, is a soft actuation. It's a soft actuator that is wrapped around the leg. So we did a lot of computation in order to make it similar, in some sense, to the tissue stimulation that we have, not to the heartbeat, to the tissue stimulation that we have in the heart. We characterize it to generate that kind of signals. This was the experimental setup. So the two mice with our wearable soft haptic device on the right leg. Both of the legs have been injected with the cancer cells. After 1 week, we wait that the cancer cells were growing. Then we stimulated 2 hours in the morning and 2 hours in the afternoon the mice for 2 days, and look to the results that are really, really interesting in my opinion. These are the results of the slice of the control of the leg. So, of course, the animal has been sacrificed, and we took this slice of tissue in the control leg, and that's the area of the tumor, okay, that was growing normally, okay, in some sense. Look what happens to the treated leg with our technology. The treated leg with our technology shows a dramatically reduced level of tumor, of cancer cells in that tissue because of the stimulation, and I will go in a moment there. The proliferation rate is going really, really down because of this kind of stimulation, and also the volume is reduced in these mice models. You see the reduction of the tumor mass from before the stimulation and after the stimulation. This is really impressive and is giving us a lot of energy to work around this concept. Also, in terms of metastasis, we went to see if this kind of cells were going somewhere else. This was not the case. We analyzed all the other organs of these mice, and now some science behind, because this was really experimental in the sense that we went to see what was going on with our wearable haptic device on the mice. But now, of course, we are asking why this happened, why this can happen. And we started another kind of research that is on the multi-wells, where we have these cultured cells of tumor with the normal tissues, and we stimulate with the same pneumatic technologies, but now designed to work on a cell. Indeed, and this is the experimental setup with the microscope to see what is going on in the culture cells. You know what can happen? So it's something completely different from our perspective of roboticist. But something similar is there. Something similar is there. Because talking with this mechanobiologist, because it's mechanobiology there, so we want to see what's going on with the tissue stimulation from outside to the cell that has the tumor inside. It can happen something like this. So the tumor cell has a mechanosensor. This is an hypothesis. They try to see if there is room between cells in front of it, okay? If there is room between the cells, then it proliferates, okay? And the tumor cells grows. Of course, if there is no room, the tumor cells does not proliferate. It would be stupid to grow in no space, okay? By having this stimulation, since the mechanosensors has some, is not a continuous sensing, is taking signals discretely, so basically we get some confusion on it probably, and it registers no space because of the stimulation, and just reduce a lot the proliferation rate because it thinks that there is no space, but the space is there. Of course, we cannot reduce, and this of course does not happen to regular cells because to regular cells does not proliferate, so they don't care about this kind of signal. Only the tumor cell proliferate, and they get the signals that there is no room inside. And of course, this kind of mechanical stimulation through the tissue and our technology allow the tumor cell to reduce the proliferation rate and the regular cell to live without problems because they, in some sense, breathe. So this is something really interesting because it right now we tested it on almost 30 animals, mice, with the different tumor cells injected, including melanoma. And we are also trying to understand, of course, the pneumatic technology was clear because the mice was small. But if now we are going towards large animals and then humans, and we will get back to the technologies that we were showing in my talk and also in the talk of Alison and Catherine before. If this keep working, probably this will open a new area of research within haptics and a lot of wearable haptics, of course, and a lot of ideas can come. And I hope to bring this area in our community in the next months. Thank you.
Thank you very much, Domenico. Very exciting perspective for haptics, actually. Questions?
Very interesting talk. Thank you very much. And a very nice application also of the technology.
Thank you.
I have a question. Just curious, would electrostimulation or physical exercise would have a similar result in your opinion?
Yeah, we tested it with electrostimulation in the muscle. We injected the tumor in the muscle of the mouse before doing our technology, and there are results with the we stimulated the muscle in that way. But it's only for the muscle. For example, melanoma is skin cancer. And it's very frequent, so you cannot get with the electrostimulation because the muscle behind the derma is not enough it's a sort of compression that we have to generate. So that's an important question, but for vascular activation it can be. So then we were testing a lot, we were doing a lot of research, for example. We were trying to see if people working on the road, like they have less incidence of tumor, to see if there is this kind of correlation, but it's still, and we are also doing a lot, I mean, together with this doctor, especially in the multi-wells genetic studies, because we want to study the mechanobiological system and also the gene, because in the future we would like to combine physical stimulation with our technology with drugs to facilitate the physiology and the process while this works. And for us, it's very important because we have to understand how from external stimulation, we can go to which is macro. We are not doing something very, precise. The reason is more a matter of choosing the frequency and the intensity that reaches the tumor cells and generate this effect. So it's interesting from a robotics point of view, more than mechanobiology. Biology explained reason, but we need to do the mechatronics behind this idea if it works.
Thank you. Thank you very much. Is there a very quick question? There.
Hi, great talk. My question was just if you've looked at injecting versus naturally occurring tumors, if there's any effect of the mechanobiology of the tumor itself that maybe is changed.
We did 2 tests. The first one was to inject lung cells in the muscle, and this was not very, natural, ecological. But then the last ones is with the melanoma, and is very, very similar to what happens normally. I don't see where you are, but I'm looking. So melanoma is much closer to the real situation, and we are working with that. But they have, I'm only bringing the mechatronics and the idea of the roboticist, especially in interpreting the signals of the cells. But all this stuff in terms of mechanobiology is done by the medical and biologists collaborating with us.
Thank you. Thank you again.